structure and shape to perform the specific function. Development. Development is the series of the changes in which organisms undergo in their way from the embryonic state to maturity, that is, from lower state to higher state of organization. Plants undergo different pathways in phases of life in order to form different kinds of structure. This is termed as plasticity. If different shaped leaves present on the same shoot or on different shoots of the same plant, it is called heterophyly. For example, water crowfoot, water startwort. Plant growth regulators. Characteristics. In all the plants, there occurs minute quantities of certain chemical substances that regulates growth and differentiation. These are called growth regulators. A growth regulator may be defined as an organic substance produced naturally in plants controlling growth and other functions at a site remote from its place of production in minute quantities. These are auxins, gibberellins, cytokinins, ethylene, and abscisic acid. The discovery of plant growth regulators. In the last 60 years, a large number of growth regulators have been isolated from plants. The first indication of their existence was given by Darwin, 1880, who was studying the bending of the coleoptile of a grass towards light. He found that the light falling on the tip of the grass coleoptile causes some influence. This influence is transmitted downward and causes the coleoptile to bend towards light. F.W. went isolate auxin from the tip of the coleoptiles. E. Kurosawa reported the appearance of symptoms of Bacony disease of rice seedlings is uninfected rice seedlings when they were treated with sterile filtrate of fungus Gibberella fujikuroi. Gibberellic acid was later isolated from the fungus. The discovery of plant growth regulators. F. Skoog observed that callus proliferates in the tobacco plant only if, in addition to auxin, the nutrients are supplemented with extracts from the vascular tissue, yeast. Skoog and Miller later identified cytokinesis, a promoting active substance, and named it kinetin. 3 independent workers reported 3 different kinds of inhibitors in plants: inhibitor B, abscission 2, and dormin. Later, all the 3 inhibitors proved to be chemically identical and name it abscisic acid, ABA. It was found that volatile substance being released from ripened oranges leads to the ripening of unripened bananas, and that was confirmed by Cousins. Later, this substance was found to be ethylene. Physiological effects of plant growth regulators. Auxins. Indole acetic acid is the main natural auxin. Indole butyric acid is also isolated from the plants. Examples of synthetic auxins are naphthalene acetic acid, 2,4-dichlorophenoxyacetic. All these auxins are very useful in agricultural field. Physiological effects of auxin on plants. Suppression of lateral buds by apical bud is called apical dominance, and it is found due to transportation of auxin. Auxin stimulates cell division in cambium as well as in the primary meristems. Cell elongation is an important role played by auxins. Auxins have the differential action on shoot tip and root tip. Induce root formation at low concentrations. Prevention of leaf falls by suppressing the formation of abscission layer. Seedless formation of fruit is induced by auxins. It is also termed as parthenocarpy. Gibberellins. Gibberellins come under the category of plant growth regulator. Physiological effects of gibberellins on plants. Gibberellins cause stem elongation and leaf expansion but have no effect on roots. Gibberellins promote seed germination in lettuce, cereals, etc. Plants like pea, bean, tomato, pepper, cucumber, lettuce and cabbage on treatment with gibberellins develop broader and elongated leaves. Gibberellins break dormancy of buds and tubers, but in root tubers, the application of gibberellic acid inhibits the development of root tuber. Gibberellin causes parthenocopy in tomato, apple, etc. Cytokinins. Physiological effects of cytokinins on plants. In the presence of auxins, cytokinin stimulates cell division even in non-meristematic tissues. In parenchyma tissue culture, mitosis is accelerated when auxins and cytokinins are present. It breaks the dormancy of many seeds and promotes their germination. It also promotes the growth of lateral buds. Cytokinins delay the process of aging in plant organs by controlling protein synthesis. It also helps in accumulation of salt in plant cells. Ethylene. Physiological effects of ethylene on plants. It modifies growth by inhibiting the elongation of stem and roots. It inhibits the growth of lateral buds and causes apical dominance. It accelerates the abscission of leaves, flowers, and fruits by formation of abscission layer. It retards flowering in most plants but induces flowering in pineapple, mango, and many other plants. It increases the number of female flowers and fruits in cucumber plants. Abscisic acid. Physiological effects of abscisic acid on plants. It acts as a stress hormone as it stimulates the closure of stomata. It inhibits shoot growth but does not have much effect on roots. It induces seeds to synthesize storage proteins. It also increases the tolerance of plants to different kinds of stress. It acts as antagonist to gibberellins. Photoperiodism. The ability of plants to respond to the relative length of day and night to which the plant is exposed is called photoperiodism. The flowering response of the angiosperms falls into three categories. Short day plants. These plants initiate flowering when the length of the day becomes shorter than a certain critical length. If these plants expose for the length greater than the critical length, then they will remain vegetative. Example of short day plants are Cosmos, Dahlia, etc. Long day plants. These plants induce flowering when day length exceeds critical day length. Example of long day plants are barley, sugar, etc. Neutral day plants. These plants flower regardless of the exposure of light. Example of neutral day plants are sunflower, tomato, etc. Vernalization. Vernalization is a process in which plants expose to low temperature in order to increase the flowering. Many temperature plants have the requirement of vernalization in order to initiate or accelerating the flowering process. Many plant species include some ecotypes of Arabidopsis thaliana, and winter cereals such as wheat must go through period of winter. Many biennial species have the period of vernalization, which can vary in temperature. Did you know? Lateral root develops from pericycle. GI3 induces the mRNA synthesis for alpha amylase enzymes in allurone cells. Summary. Let's summarize what we have learned. An irreversible increase in the size and number of cells by division and enlargement is called growth. Three phases of growth are meristematic, elongation, and maturation. The increased growth per unit time is termed as growth rate. The conditions necessary for growth are nutrients, water, oxygen, temperature. Differentiation is the process by which meristematic cells undergo changes in the structure, shape to perform the specific function. Dedifferentiation is the process by which the already differentiated or mature cells get back the power of division and become meristematic. Redifferentiation is the process by which the meristematic cells formed by the dedifferentiated cells undergo changes in their structure and shape to perform the specific function. Growth regulators are auxins, gibberellins, cytokinins, ethylene, and abscisic acid. The ability of the plant to respond to the relative length of day and night to which the plant is exposed is called photoperiodism. Vernalization is a process in which plants expose to low temperature in order to increase flowering.